Good morning. I would like to thank the Sociedad Argentina de Neurociencias, especially Dr. Diana Jerusalinsky, for having organized this satellite symposium. I will talk about our experience in Buenos Aires about the use of biomarker in dementia. I would like to describe some subjects. First, the discussion about neurodegenerative disease or cognitive proteinopathy. Second, I will describe the clinical use of biomarker in dementia. Third, I will talk about our experience in Fleni with biomarker. Fourth, I will describe the new stage for Alzheimer's disease, because Alzheimer was synonymous of dementia, but today we well know the pre-dementia state of Alzheimer's disease. After that, we will talk about the clinical use of biomarker and the new classification, the new ATN approach to Alzheimer's disease. And finally, our conclusion. Some words about the discussion between neurodegenerative disease and proteinopathy. At the beginning, the description of neurodegenerative disease, Alzheimer's disease, frontotemporal dementia, Lewy body disease, among others, were diseases that produce degeneration in selected systems from the brain. Motor extrapyramidal system in Parkinson's disease, cognitive network in Alzheimer's disease, behavioral frontotemporal network in frontotemporal dementia. But different phenotypes could produce but the same neurodegenerative disease. As example, we have Alzheimer's disease with different clinical type or phenotype. Amnesic phenotype, posterior cortical atrophy, primary progressive aphasia, and even frontal variant of Alzheimer's disease. Other example is frontotemporal dementia. We have different phenotype of frontotemporal dementia, like, as example, the behavioral variant, the aphasia variant. And also, we can, phenotype could be produced by several diseases. One phenotype, several diseases. As example, a progressive primary aphasia could be due tauopathy, TDP-42, or even Alzheimer's disease. Each proteinopathy could determine several clinical phenotypes. Then, as you can see in our last paper in Dementia and Neuropsychology, we say that we must replace the word where by the word what. Now, we talk about the biomarker in dementia. What are the biomarkers? Biomarkers generally refer to a measurable indicator of some biological state or condition. The change in different proteins can be determined candidate for biomarker, and we have biomarker for DOPA, for amyloid, as example of biomarker for amyloid if the protein A beta 42 in CSF, the PET with amyloid, biomarker of tau, the phosphotau in CSF, or the tau marker in PET scan, biomarker of neurodegeneration like the atrophy on MRI scan, the total tau in CSF, or the FDG metabolic PET, or the biomarker, the genes, the different genes that can be biomarker. Okay, now I will describe our experience with the biomarkers. This is our first paper published in 2013 with our first result with A-beta 42, total tau, and FOFO-tau in CSF in a follow-up of 4.7 year in patients with mild cognitive impairment that progress and not progress to dementia, patients with dementia of Alzheimer type, and frontotemporal dementia. The next slide, we can see our first amyloid PET in 2012 in one of our patients with Alzheimer's disease. You can see the amyloid deposition in red in frontal cortex and precuneus. This is the typical image of Alzheimer's disease. Next, in 2011, we become part of ADNI project because we like to be included in the adaptation and standardization of the biomarker. The ADNI, the Alzheimer's Disease Neuroimaging Initiative, is a multi-site protocol that work with all biomarkers of Alzheimer's disease. We launched the Argentina ADNI and we organized the management committee and 4 streams, 1 clinical stream, 1 laboratory stream, 1 neuroimaging stream, and the epidemiological stream. Based on ADNI, we developed a clinical platform, a biochemical platform, and a neuroimaging platform. And also, we developed the brain bank of the institution. Our biochemical platform was organized by Dr. Ezequiel Surace, and each year we undergo the quality control program of Alzheimer's disease biomarker from the Alzheimer organization. We perform in CSF, A-beta 42, phosphorylate tau, total tau, and neurofilament. As example, I would like to show our paper with our result of CSF neurofilament as neurodegenerative biomarker. Most interesting is the second paper. This second paper was from Diane. Diane is one network, dominant inherited Alzheimer network, with patients where we study the neurofilament in serum, and we found this biomarker of neurodegeneration 10 years before the symptoms. And this is very interesting because it's an easy and cheap biomarker. This is very, very interesting for the clinical use in the next year. This is our Center of Molecular Imaging at Fleni, where we perform the PET scan with FDG for metabolic study, amyloid marker, or tau marker. We have also the cyclotron to produce the biomarker. Nowadays, we have over 900 amyloid PET scan performed, no? We can see on right the FDG from one Alzheimer patient with the hypometabolism, parieto-temporal, and in the middle we can see the amyloid PET. The last is the tau PET with the marker in the temporal lobe. This is very interesting to study the Alzheimer patient. Finally, as part of our multimodal approach, we have a brain bank with approximately 100 brains. This is one of our paper about one of our Argentine families with presenilin 1 mutation and the neuropathology. Now we will talk about the different stage of Alzheimer's disease. We will talk about the pre-dementia state. This slide is part of our collaborative work, DIAN, Dominant Inherited Alzheimer Network. On left, we can see the amyloid PET, the metabolic PET with FDA, FDG, and the study of cortical thickness by brain MRI. On right, we can see the presence of the different biomarkers before the onset of the symptoms, 20 years for amyloid and 15 years before for neurodegeneration biomarker, FDG and cortical thickness. This is very interesting because we study the next generation of the patient. We study the asymptomatic patient, and we found this interesting information about the beginning of the disease. Now, we will talk about the new clinical approach called ATN, the new clinical classification for Alzheimer's disease, amyloid, tau, and neurodegeneration. On left, we can see the paper from Clifton Jack with a description of the ATN. We launched this new classification scheme for Alzheimer's disease with biomarker. On right, you can see our two last papers with the use of the ATN in our Argentine cohort of ADNI patients. In this slide, we see our result: normal control, early mild cognitive impairment, late mild cognitive impairment, and dementia of Alzheimer type. In red, we can see the positive amyloid patient, in gray the negative ones, and in blue the SNAP patient. And we can see this distribution in each population: normal MCI, late MCI, and dementia patient. And you can see on right the ATN classification. The next slide is the same study where we perform two survival analyses, one with the clinical phenotype on left and the other on right with a multimodal approach with biomarker. You can see that the right one is the most interesting because the discrimination is greater. Then the conclusion. And I published 8 years ago one paper in the Journal of the Neurological Society, the Argentina Neurological Society, and I say, you can hide the sun with a hand. New diagnosis approach with biomarker for Alzheimer's disease is the challenge of diagnosis for the next generation. Thanks again, Dr. Diana Jerusalinsky, for the invitation.